All right, so the next speaker is Jonathan Weissman. Dr. Weissman is a Howard Hughes Medical Institute investigator and a professor of cellular and molecular pharmacology at the University of California, San Francisco. He received his undergraduate physics degree from Harvard College and his PhD in physics from MIT, where he worked with Peter Kim in the protein folding field. Following his PhD, he moved to Yale University for postdoctoral training in Arthur Horowitz's lab, and he worked there on studies of GROEL and GROES. Dr. Weissman is a member of the National Academy of Sciences and a fellow of the American Academy of Microbiology. And his numerous honors include the Raymond and Beverly Sackler International Prize in Biophysics, the Keith R. Porter Lecture Award from the American Society of Cell Biology, and the National Academy of Sciences Award for Scientific Discovery. And today he will speak to us about work in his lab that's focused on CRISPR interference and CRISPR activation, which give us the ability to turn any desired gene on or off. So please join me in welcoming Dr. Weissman.
Thank you so much for that kind introduction, and for the Gardner Foundation for putting on such a lovely event, and of course inviting me to speak. So, as we all know, every cell in our body has more or less the same complement of DNA, yet we're able to have this huge diversity of cells, at least 200, now people think maybe 2,000 or more different types of cells. And the way this is possible is that each cell is expressing a different complement of the sets of genes in its DNA. And of course, this control of gene expression is important not only for allowing the diversity of cell types, but also a critical role both in disease and even in evolution, where the difference between the proteins coding regions of us and a chimp are really very minimal. So much of the changes we think have to do with just which genes are turned on, how much, and when. Another remarkable thing that's come out from sequencing the human genome is that we only have about 20,000 or so different protein coding genes. And so it's on the face of it, seems surprising that you can get this enormous diversity. Of course, the answer that we all know is this is possible due to combinatorics. So just in the same way that you can have a piano keyboard with only 88 keys, and yet depending on the score, which notes we play, how loudly, and in what cadence, you can play anything from Mozart to Ray Charles. So over the last 20 years, thanks to things like microarrays and RNA-seq or ribosome profiling approach my lab has worked on, we've been able to listen to this music with increasing precision. So to be able to quantitatively monitor which genes are turned on, and to do this now even on a single cell level. And that's been enormously fruitful on one hand. On the other hand, it's not completely satisfying. It lets you make an observation and try to associate it with, say, a disease or change in differentiation. But really what we'd like to be able to do is go beyond simply listening to this music to compose our own scores. In other words, to be able to control at will the expression level of any gene or any set of genes. And this has now become possible, and I'm sure you all know the answer of why, which is through the technology developed from CRISPR-Cas9. In particular, a critical moment in this was the discovery that a single protein, Cas9, and then a single chimeric guide RNA would make a programmable endonuclease in which we could program where the cleavage was occurring by the simple rules of base pairing of our guide RNA to the DNA. And this has been, as we've heard, an enormously powerful tool for genome engineering. But for our purposes, it actually didn't really do what we wanted to do. So this tool would let you, in the analogy of the piano, would let you fix a broken key or break a key that's working, but doesn't allow you to sort of, in a reversible, controllable way, to hit the different notes. So in order to do this, we had to make one modification, which was to take this beautiful endonuclease and break the scissors. And that was possible because we knew what the catalytic sites were. And so a simple point mutation would turn this from a programmable DNA binding, programmable nuclease rather, to a programmable DNA binding protein. And I should say that we here, the really critical person in making this so-called dead Cas9 was Stanley Chi when he was a fellow at UCSF, who's now, and has been a critical collaborator for much of the earlier work we've done in mammalian cells using this. So now you still have a programmable binding event based on base pairing between the guide RNA and the DNA, but this reaction stops there. And what makes this so useful is you now can put a fusion with the dead Cas9 protein and bring in different effector domains. And we and others have now brought in a wide variety of different ones, but the two that I'm going to talk about are either ones that inhibit transcription, so for this CRISPRi approach that will let you surgically turn off a neighboring gene in a reversible way, or bring in activation domains that let you turn on the neighboring gene. But as I said, we and others have now put in either fluorescent proteins or a wide variety of different epigenetic modifiers. So in a sense, one can think of, if you think of the CRISPR-Cas9 as being a very powerful tool for editing genomes, the dead Cas9 basically allows us to do an analogous thing, but to edit the epigenome. So in essence, we now have a volume switch where we control the expression of any gene. So having done this for one gene, of course, we want to do this for all genes. And thanks to sort of screening technologies that have been worked out, that was relatively straightforward. And here I'm showing you an example of genome-wide screens for growth, both for the effects of CRISPR interference shown here. So genes down here are ones that are, when knocked down, are causing cells to become sick, or for CRISPR activation. And so these genes are ones that, when you overexpress, cause the cells to become sick. And there's sort of a few take-home messages from this. First, so every gene is plotted by its CRISPRi and CRISPRa score. What you see is that they're highly orthogonal information. So what you're getting at, the genes that when you knock down are causing cells to become sick, are really distinct from the ones that we overexpress that are causing this phenotype. The second is there's a lot more genes that when you knock down cause the cells to become sick. But if you were to put the labels on those genes, what you'd see is, by and large, these are generic housekeeping genes, things that replicate the DNA, synthesize, transcribe RNA, synthesize proteins, or basic metabolic processes. By contrast, the strongest hits on the CRISPR activation tend to be much more cell type specific. And actually, among the top hits, what we're getting are known tumor suppressor genes. And that makes sense, because a tumor suppressor gene is a natural break. So if you overexpress the break, the cells can't grow. And this is, when graphed like this, each of these seem fairly dry, but when you actually go and look at the cells, they're often doing rather spectacular things. So here's an example of what happens when you overexpress a polo-like kinase, and you see you get the spindle, a huge number of spindles and excess replication, and it's very clear to see why this cell is not going to be able to divide rapidly. So these were examples from really our first genome-wide screens. Since then, we, and really this has been work led by a terrific student in my lab, Max Hurlbeck, have done many different screens, and from this use sort of standard machine learning type algorithms to try to find out what were the rules that made a guide effective versus non-effective, to get a much better tool out. And by and large, so these rules have come in two forms. One that has to do with the sequence of the guides themselves. And others have done this with CRISPR cutting, and I have to say, we've largely converged on the same type of answers, not having homopolymers, not having Pol3 transcription inhibitors, et cetera. But what Max was able to do was to get a second orthogonal access that was critical to really turning this into a high precision tool. And that came when Max plotted the efficacy of his guide RNAs as a function of distance to the transcription start site. I should say, so this is the genome-wide data that Max collected. This is phantom data collected, of course, in Japan, completely independently. Yet you see this really remarkable pattern that was coming up. And the identity of the pattern became clear when we took a third genome-wide data set, that from the nucleosome positions from the ENCODE project. And what you see here is that the guides that are active are exactly out of phase of where the nucleosomes are. And of course, this makes a lot of sense. CRISPR-Cas9, as wonderful as it is, never evolved to deal with nucleosomes. And so if you have a highly occupied nucleosome position, you're going to have a hard time. It's going to act as an inhibitor of either the binding or the cutting. But by incorporating these two rules, both the sequence of the guide and the underlying sequence chromatin structure of the DNA substrate that you're targeting, Max was able to greatly improve the libraries. So the sort of acid tests for these types of libraries are something as precision recall. Basically, the community agrees on a set of genes that are really likely to be essential, and then a set of genes that are really unlikely to be essential. And then you do a genome-wide screen, and you say, using a scoring, how well am I able to identify the essential genes while avoiding the non-essential ones? And you can see for our V1 library with 10 guides per gene, we were able to get about 80% of the essential, and then it sort of fell apart. With the V2, we're now able to go to about 95% before we start to get any significant false positives. And we actually think the last 5% may be just sort of errors in what we think are the non-essentials. But what was even more remarkable and important from a technical point of view is Max was confident enough of his scoring that he would split the library into two, his 5 best and then the next 5. And so we could then say, what if we just did 5 guides per gene? How much would it deteriorate? And the answer is it basically did exactly the same. So now we have compact, highly effective guides. We think something like 80% of the guides in this library are highly effective at knocking down the gene. And by highly effective, I mean sort of typically able to get 90+%. And although this was all done in our sort of go-to cell line K562, he was able to show that this actually had similar efficacy across a wide variety of cell lines, including human iPS cell lines. So we now have CRISPRi and CRISPRa libraries, genome-wide libraries in sub-libraries, both in human and mouse, and they're all available on AdGene for the community. So with this type of libraries, we can define the relationship between expression and phenotype. So here's an example where phenotype is resistance to ricin, and either through CRISPRi or CRISPRa, we've changed the expression level over about 1,000-fold, and you can see how this is affecting the ricin phenotype. We can do genome-wide forward genetic screens for any transcript, whether it be essential, non-coding, or non-essential protein coding. You can do overexpression screens, and this is particularly valuable for things like drug resistance or to get phenotypes for redundant genes or other gain of function. And we can do massively parallel combinations. So at University of Toronto, I should hardly have to espouse the value of doing genetic interactions with the beautiful work from Charlie Boone and colleagues in yeast and showing how powerful a tool this is for functional discovery. But basically, we can now do the same types of things in mammalian cells, and actually, because it's a pooled format, we can get data actually much more rapidly and arguably with higher precision. But it's in addition to this discovery of mapping out protein function and mapping out pathways, this type of synthetic lethal is also a very powerful way to look for a combination of drug targets that will give us much more specificity and efficacy than we could get from anyone alone. And that's particularly interesting and of interest in the cancer field. So what I'd like to do now in the rest of the talk is discuss 2 applications of this CRISPR-based screening approach. The first is a systematic definition of the function of these long non-coding RNAs. And then the second is a very new technology that lets us get much richer phenotypes than just life-death or turning on a reporter gene. So first, I'll start with the CRISPR-based screens of lincRNAs. So one of the big surprises to come from the Human Genome Project, sequencing transcripts, was that there were many RNAs, messenger RNA, the RNAs that looked like messenger RNAs. They were capped, they were polyadenylated, they were often long by definition, in this case greater than 200 base pairs, but they had no parent protein coding potential. And they were, and basically by phylogenetic analysis, you could really have high confidence that there was no at least conserved protein coding component. Well, a number of these links have been studied in exquisite detail. So for example, the excess RNA that's responsible for X inactivation, or the TERK, the RNA that the telomerase is using to guide production of telomeres. Very few of these tens of thousands of non-coding RNAs have been studied in any detail, let alone assigned any function. And basically, the reason why this is the case is that we've lacked tools for surgically and with high precision inhibiting the function of these lincRNAs. So unlike a protein coding gene where you could make, for example, an indel, and then the gene is out of the protein coding frame is disrupted, and you know that you've disrupted the protein, we don't know which parts of the lincRNA are functional, so there's no quick way to inactivate them. But we reasoned that the CRISPRi approach, by being able to precisely turn off a lincRNA, now gave us an opportunity to do this. So we, being Max Holbeck, who developed the CRISPRi libraries, and a partner in crime, John Liu, who's a graduate student in Dan Lim's lab, have been working on this for the last year and a half or so. They developed a set of libraries they called the CRINCL libraries for CRISPR non-coding libraries. It's 17,000 links to targets and about 170,000 guide RNAs. And these were targeted against 7 highly diverse cell lines. 6 of them transformed, 5 of them cancer cell lines, and then the 7th is a human iPS cell. And basically, through massively parallel oligosynthesis, production of lentivirus, in which each lentivirus is encoding a single guide RNA, then infecting in a pool, so each cell gets a different guide RNA, and then growing to see that to monitor the effect of knocking down the lincRNA that's targeted by that guide. You can then, in an automated way, get a phenotype where if guide RNA is targeting an essential link, you'll see it drop out of the population, and if it's non-essential, you won't see it. And from this, in this pooled manner, you can basically get a growth phenotype for all of the lincRNAs, and to do this in a single experiment. So here's a little bit of the more raw data from human iPS cell lines. We use these so-called volcanopots. Basically, this axis is telling you the growth effect. So the further you are over here, the bigger growth effect. And this tells you the statistical significance. And so all of these here are hit genes that are well outside what we would get from our internal negative controls. These are highly reproducible from when we do full biological replicates, and when we clone out the guide RNAs and test them individually, they also almost invariably retest. So each of these is really a high confidence hit gene, hit lincRNA that when we knock down, will give the phenotype. Across the cell lines, we were able to identify nearly 500 lincRNAs that modify the robust cell growth rates upon knockdown. So this is vastly increasing the number of lincRNAs for which we have a phenotype, giving a tool for knocking down that link to study it in depth, as well as identifying the cell line in which it has this phenotype. The surprise came, though, when we looked at the data in terms of the specificity for the cell type. Of those 499 lincRNAs that had phenotypes, precisely 0 were common to all cell types, and the vast majority were common only to a specific cell type. And that was interesting because analogous work on protein coding genes, for example, the work from Tim Wong in David Sabatini and Eric Lander's lab, had found really very much the opposite effect. As I said, most genes that have a growth phenotype, when knocked down, tend to be housekeeping proteins, like RNA polymerase or translation. And only a relatively small fraction would be specific to one cell type. And so that's, I think, really interesting, because what's this saying, when we go back to that first slide I showed of the baby with all the different cell types, is that the lincRNAs are playing a critical role in giving identity to these different cell types. And so that's really interesting from a biological point of view, of course, because we're interested in how this diversity of cell types are possible and are maintained, but also even from a therapeutic point of view. It's much more desirable to think of a target that would be specific to a cell type and even specific to a cancer cell type than a generic housekeeping protein that would cause broad growth defects if targeted. All right, so that's sort of the end of the link part. And I'm going to now switch to a very new technology that we've just completed. It's been a wonderful collaboration with the lab of Aviv Regev. And that is our effort to go not from just watching a growth defect, like when you knock this gene down, does it cause cells to grow faster or slower, but to get a much richer phenotype. And in general, through these types of screens, there's been a sort of a compromise. You either look at many genes and with very simple phenotypes, or you study the effects of turning on and off one gene and in great depth. And the technology, we call it a Perturb-seq approach, allows us to marry this, both to do pooled screens with many different genes at once, but to get very rich single-cell RNA-seq readout. What made this possible has been the development, just in the last year or so, of droplet-based single-cell RNA-seq approaches. And in these approaches, one creates a so-called inverse immersion in which you get a water droplet that contains a bead with an oligo-DT, barcoded oligo-DT, so the oligo-DT, so you can prime off the poly-A RNA, and the barcode, so it tells you which cell you're in, and a single cell. So you get these droplets that have the cell and then a way of barcoding all the RNAs in those cells. And what makes this so powerful is you can make tens of thousands and 100,000s of these droplets rather rapidly. That one then can pool all this and do massive sequencing. But when you sequence, because each RNA has a barcode that tells you which cell it came from, you're able to assign the transcription profile to each of the individual cells in these tens of thousands of droplets. So what we did here on top of this to make the Perturb-seq approach possible is to infect each cell with a different type of guide RNA targeting a different protein coding sequence, and to develop an encoding strategy so that when one did this single-cell RNA sequencing, not only did you get the perturbation, what the effect of knocking down the gene that you were targeting was on the RNA signature, but you also read a separate barcode that told you which guide RNA was being expressed and therefore which gene was being perturbed. And in this way, you get this massively parallel data set that's giving you the gene that's causing the perturbation and the phenotype of that perturbation in terms of this very rich single-cell RNA-seq data set. The problem that we have focused on, the biological problem that we've chosen to apply this to, is protein folding in the endoplasmic reticulum. So all proteins in eukaryotic cells that are secreted or on the plasma membrane first go into the ER in unfolded form. The ER, and then only after they fold are they able to get to the cell surface. The ER is basically a dedicated protein folding vesicle, and in some cells, like a professional islet beta insulin secreting cell, they're basically the cells turn into this massive factory for making insulin, and actually sort of an islet cell can secrete the equivalent of its own mass every day. And the way this is possible is that this environment within the ER is highly specialized for protein folding. But of course, like any process, this goes wrong, and the cell has an intricate set of sensors commonly called the unfolded protein response, that is constantly monitoring the status of protein folding in the ER, and when it sees that something goes wrong, turning on transcriptional responses. And of course, this was rightly honored by the Gardner Award in 2009 to Kaz Mori and my colleague, friend, and collaborator, Peter Walter at UCSF. In mammalian cells, there's 3 distinct branches of the unfolded protein response. And each one has its unique and in some ways even rather bizarre way of turning on its sets of genes. So they all have some sensor in the lumen of the ER that's asking what's happening in the ER. The XBP turns on IRE1 turns on transcription by a totally non-conventional, totally unprecedented mRNA splicing event. ATF6 does this by a proteolytic event that releases a transcription factor on the ER surface, allowing it to go to the nucleus. And then PERK does this by actually a translational control, which then paradoxically allows unique translation of a transcription factor called ATF4, and then downstream and CHOP and transcriptional response from this. We also know from work from Peter and others that this system is constantly making a decision. Should it repair and try to restore homeostasis? But if that's not possible, it then induces a killing program, an apoptosis program, that's presumably there to say if the cell has gone to the point where it's unrecoverable, it's better that it die and the rest of the organism survive. So while there's been beautiful work in defining the mechanism of these and understanding what they're sensing, we really have very little idea of why are they these 3 different branches? What different things are they sensing? How are they being used in an orchestrated way to allow the cell to respond to the wide variety of different challenges and to make these decisions between repair and death? So we thought this was a terrific first application for the Perturb-seq approach. I'm really just going to give you a snippet of the data for time and just because I wouldn't be able to do justice to it. But just to give you an idea of the scale, for this we did 100 different genetic perturbations, although that could readily be scaled up. 500 single-cell experiments per perturbation, and that's about 50,000 cells per library. And to give you an idea of what was involved in this experiment, using a 10x, now the commercial devices for doing this, it's about 10 minutes of machine time. The library prep was about a day. It was about 2 days of sequencing, and then about 6 months of data analysis. And by far, the least complete part of this is the data analysis. And in fact, Tom Norman, this is work of Britt Adamson and Tom Norman, 2 fantastic postdocs in the lab. The mathematics that Tom used to analyze this data was things that has been developed just in the last 5 years by this guy, Terence Tao, who's sort of arguably the greatest living mathematician. As an aside, he wrote a fast algorithm that made it possible to do the decomposition that was critical for getting a strong signal out of these noisy single-cell data. But I'm going to talk about 2 vignettes from 2 of the 100 single-cell experiments. One about what happens when we knock down a key chaperone called BIP, or HSPA5, and the branching bifurcation at the level of individual cells, and the other about what happens when we knock down the Sec61 translocon. So here's results from the BIP knockdown. This is a typical way of showing single-cell experiments where you take these very high-dimensional space and project it down to 2 dimensions that maximally distinguish between different cells. Each of these dots is a cell. Over here are the control cells, and then up here are the ones that have been knocked down in HSPA5. And you can see that they readily distinguish between each other. But you can also see that there's different classes of responses among the cells that have knocked down in HSPA5. And that became even clearer when Tom ordered the cells based on the level of this response. So each of these is the 500 cells, and then looking at the degree of activation of the 3 branches of the unfolded protein response. And what he sees is that there is this bifurcation, cells making different decisions. So a subset of cells really just only turn on this ATF6 branch. And then Another subset are turning on both the IRE1 and the ATF6 branch. And then in a switch-like manner, really all or none manner, a smaller subset go all out and turn on all three of these branches, and those presumably are the ones that are now going into the apoptotic pathway and committing themselves to death. And you might think, well, this is a single-cell analysis. Maybe the CRISPR was working better in some cells than in others, and the ones that was really knocking down HSPA5 are the ones that are turning on all of the alarms, and the ones where it's barely knocking down are just turning on the ATF6. But because it's a single-cell experiment, RNA-seq experiment, we can actually measure the level of HSPA5 or BIP knockdown in each of these cells, and we can distinguish those with different response. And basically the answer is there's no differences. And the differences in the cell response is not due to differences in knockdown of the HSPA5 or BIP. It's due to an intrinsic difference of decision of bifurcation that's occurring at the cellular level, and one that we're now very interested in trying to understand both the mechanism and the physiology. But just really illustrates that if you do a bulk RNA-seq experiment, you're really going to be obscuring what's going on because you'll be mixing all these types of cells to an average response that no single cell is actually having. So the last story then is on Sec61. This is in some ways a simpler story. Here what we found is when we knock down Sec61, or actually any components of this translocation machinery that's responsible for allowing proteins to enter the ER, is that we had a robust activation of the IRE1 branch. So that's shown here with a reporter. So these are follow-up experiments, but it was very clear in the initial single-cell data. We get a very rapid and robust and prolonged activation of the IRE1 branch, but absolutely no activation of this PERK. So this was very interesting because recent studies have shown that IRE1 is actually hanging out on the translocon itself. Also, when we looked at which genes were under control of IRE1 versus ATF6 versus PERK, we saw that IRE1 was specifically turning on the translocation machinery. So now we have a classic feedback loop, homeostatic feedback loop, where IRE1 of these three branches of the UPR that are monitoring the status of the ER, IRE1 is uniquely monitoring the status of the translocon, and in return, able to uniquely repair this by turning on induction of new translocon components. And so this gives the cell a way of monitoring the status of translocation, repairing this, and doing so without setting off all the alarms and turning on PERK and inducing apoptotic pathway. So finally, I'll switch to a question which I don't have the answer to, but one that I think is worth thinking about, which is, I hope I've convinced you that CRISPRi and CRISPRa are great research tools. The question is, could these actually be used as therapeutics in the same way that people are thinking and applying genome engineering? And there's obvious disadvantages. There's a big delivery problem. Because the effects are transient, it makes the delivery problem all that harder. And if you have to have continued expression of the dead Cas9, you have a continued risk of antigenicity. However, I think there's also a number of potential advantages. In vitro, you can imagine this facilitating complex reprogramming events, for example, trying to make islet cells. And because you could do this in a way where you actually were just delivering RNP particles, not cutting any DNA, not making any permanent changes, it could in principle be, first of all, you have more sort of levers to pull by being able to turn on and off. And second, you in the end end up with a less perturbed system, so less worry about off-target effects. Again, this is because there's no introduction of foreign DNA or genome alterations. It's also been a beautiful paper from Alberto Lombardo recently that showed that if you put the right cocktails of epigenetic modifiers, you could go from these transiently turning on or off genes to actually creating a heterochromatin in a state that even by a transient delivery would allow for months of repression of a gene. And then finally, I think the transient nature of the alterations itself could be actually an advantage. And the reason I say that, the number of genes that one can safely turn off for the lifetime of a person and know that there's going to be no adverse effects is actually quite small. How would you know this would? you either have to have a human who's naturally mutant in that gene and is otherwise healthy, or you have to basically do the safety over the decades of life of a person, which is effectively undoable. So as an example, I would think of this as one might transiently turn down the receptor for a virus and then wait for a couple of weeks until the immune system kicks in and naturally clears the virus. And then at the end of the day, the person's restored to their natural the receptors there, their DNA isn't edited. So with that, I'll thank the people who did all this work. I don't have time to really do justice to all the people, so I'll just focus on a few of the key players. Of course, Stanley Chi for the critical initial work on CRISPRi, Luke, Gilbert, and Max Holbeck for developing the CRISPRi, CRISPRa approach in mammalian systems, Britt Adamson and Tom Norman for the Perturb-seq approach. It's been a wonderful collaboration with Aviv. And then John Liu and Max Holbeck and our collaborator Dan Lim on the linkRNA story. That I'll end, and thank you guys. I guess I'll take questions later.